Thank you very much for the opportunity to give this webinar today. It was a quick invite and I kind of had to move some stuff around, so I'm very appreciative of the time and appreciate that you're listening to me discuss non-coding RNAs. Let me put this up real quick for everyone. And I apologize if my slide deck isn't the best it should be, but you're going to get a dose of a little bit of understanding how non-coding RNAs work, what we're kind of interested in studying with respect to the expression pattern of non-coding RNAs in the brain, and how we're going to, or attempting anyway, to uh, develop biomarkers for early detection of certain types of brain cancer, but mostly turn some of these things into actual therapeutics. So,
Dr. Adams, I'm so sorry. Are you sharing slides currently?
um, uh, hold on one second.
Okay, just making sure. Thank you.
Yeah, my computer's a little slow. Uh, All right. So, I don't know. I mean, for the audience in particular here, how much everyone really knows necessarily about either non-coding RNA or more formally brain cancer. So I just really just put up a few slides here, and I kind of open the door for more of a discussion-based session if there's needs to be. Um, so thank you for the introduction. Basically, I founded my company back in 2016, 2017 with the goal of developing biomarkers, like I said, and therapeutics for various types of brain cancer, the predominant one being glioblastoma. And the rationale for that is, and I understand that this is still a bit of an older slide back from 2005, the journal, New England Journal of Medicine, a study done a few years back. But unfortunately, the outlook for those individuals diagnosed with glioblastoma is still a pretty stark diagnosis. Depending on if you're getting a combination of radio or TMZ treatment, you're still somewhere around 50% survival rate. Um, Depending on what kind of modality you're in, usually a few years. And so uh, this number has been quite actually stable for some period of time. I think I just went on to the SEER database and I had some of my students run some analyses, and the data is still, even though there's a little dip in some of the prognostic data, for the most part, these values are still remaining the same. And it could be for a whole host of reasons. The predominant one being really early detection of the disorder. Glioblastoma is really technically great for astrocytoma. So that is a cancer of the astrocytes themselves. So it's, by the time, as a clinical entity, it's already a metastatic or more of a widespread disorder. So that obviously creates some clinical challenges and sort of barriers for treatment. Um, but I think what has happened and what I have really gotten very interested in as one of our modalities for getting at early detection of brain cancer is really targeting the neuroinflammatory state. And this is not my review work. This is the a kind work of another colleague in the field. But we also just recently published a manuscript, a review on the non-coding RNA inflammatory state in how it can sort of promote a precancerous state. Um, but I wanted to put this slide up here because it does emphasize where the field sort of stands with respect to the widespread strategies being employed in the clinic right now to take advantage of quote unquote brain tumors from an immunological standard. And I think this is an important point to mention because I think for most of us through our academic training, for many, many years was originally taught, that the brain is sort of a neuroprotected area. It's a blood-brain barrier, that it doesn't really have. It's sort of protected from these immune system in general. So I think for a while, many researchers didn't even think to perhaps even use an immune strategy to target tumors of the brain. But clearly, from this review, you can clearly see the widespread uh, opportunities for clinical intervention. I mean, just simply with some anti-PD-1 inhibitors, which has been widely employed in many different cancer models, all the way down to CAR T therapy, myeloid therapies, and even oncolytic viral therapies. And this may seem kind of, uh, you know, uh, isolated from the field that I'm in, which is a non-coding RNA field. And you might be wondering, why would I be interested in it? And both the neuroinflammatory state. This seems to be more of protein biology, really less of non-coding RNA biology. But the two fields are actually quite linked very well. Non-coding RNAs in general, if I want to just pull up a particular pathway, are really sort of regulator of genomic expression. So it's sort of not just whether or not a gene is turned on or off or present, not present, but really the abundance of a particular protein coding gene, for instance. So in many of these disorders, what we have failed to look at early on is really the non-coding repertoire of that particular tumor. And I'm pretty sure that many others have started to look at this. I've looked at people have been invited to give these webinars, and I think there's been more of appreciation for looking at the non-coding RNA state. But my entire life has been studying non-coding RNAs, even since college, when we used to call these things antisense RNAs or antisense oligos. So from a biochemical perspective, um, you might just have simply thought that a non-coding RNA, such as a microRNA, interacts with a targeted messenger RNA through Watson and Crick base pairing, and somehow inhibits the expression of a gene product, or by preventing the translation into a protein product, or actually blocking, or sorry, degrading the message itself. So what we used to think of as another protein called argonaute can actually bind and then degrade the target RNA. But what we're finding is it's a little bit more nuanced than just sort of a microRNA interacting with a particular messenger RNA. We find situations where microRNAs can interact with other non-coding RNAs, such as long non-coding RNAs. So a few key examples of that would be like hot air, MALAT1, uh, exist for X chromosome inactivation, things of this nature. Those are pretty well published long non-coding RNAs. And what we're finding is that the interaction between a microRNA and the long non-coding RNA may influence not just the abundance of a long non-coding RNA. So you could think of a long non-coding RNA just like a messenger RNA. You could kind of regulate the abundance of that, what we would call a link RNA, but also can kind of modulate the ability of those long non-coding RNAs to act as chaperones, essentially to be grouping and bringing into proximity other RNA binding proteins, other protein machineries that could also interact with the chromatin structure itself. So you can almost have a situation where a microRNA could regulate even the transcription of certain gene products. So we're finding that there's, as has been mentioned before by others in the field, kind of a non-coding RNA language. And it sort of takes a little bit more finessing to understand and translate that particular message or that kind of language. Predominantly because for some of the main reasons is long non-coding RNAs, for instance, tend to be expressed at lower levels than most messenger RNAs. And so that can kind of be a little difficult when we're trying to infer whether or not there's a relevant long non-coding RNA of interest that maybe has some very important biological activity. We sort of give priority to high abundance things and kind of toss away the low abundance stuff. So I think from our perspective, we don't really look at abundance per se. We really try to focus on differential expression patterns and control our experiments to sort of very nuanced experimental settings where we can really tease out a specific function of the long non-coding RNA or a microRNA rather than just focusing solely on, if it's up or down regulated in a cancer or precancerous state. And then we also do function a little bit on the new thing called the circular RNA, which I don't want to bore too many individuals in this particular group about that, but there are pretty exciting molecules to study. They seem to be um, uh, sort of maybe we thought once was improperly formed biochemically, a link RNA or something like this, but there's a back splice present in the message, and we get a circular formation of an RNA. And these circular RNAs can be sponges, meaning they could be sinks essentially for certain microRNAs. So microRNAs can actually interact with circular RNAs. And just because of their sort of structure, can modulate the regulatory actions of microRNAs on a messenger RNA. So there is quite an intricate language of different non-coding RNAs, and they all kind of feedback into, in this case, regulate the abundance of what I'm trying to show on this slide, gene A, which may make a couple of different proteins. Okay. So this is probably a slide that's probably a little more simplistic, and I like to show this slide because we not only study just the abundance of these various non-coding RNAs, we also study the mechanisms at which microRNAs are generated, because that also brings into play some protein chemistry, which allows me to kind of collaborate with some others outside the non-coding RNA space. And what we've actually found, though, is that in certain uh, brain tumors, I can't really show as much of the data as some of my other colleagues, probably because I'm on the business side a little bit. But through some collaborations, and if people want to connect with me, we can kind of go down that path. But what we have actually shown in some preliminary data is some of the biogenesis machinery of the microRNA network here is differentially active, we'll say, in a cancerous state versus just what you would think of as a normal astrocyte. Okay. And I can't tell you which one necessarily, but this is kind of important because things like drosha and pasha, which is the drosha and DCCR RNA complex, basically recognize the microRNA genes that are being transcribed initially and process them into a primary microRNA transcript. And the reason why this is important is that not all microRNAs are actually made in this fashion. Some microRNAs are actually quite literally the entire intron of a messenger RNA gene. And what we've actually found quite frequently are many of these mirtrons are differentially expressed in a certain, not so much glioblastoma, but a couple other rare forms of brain tumor. And we suspect that there might be a different mechanism at play for this. And that's hopefully a study that we're going to try to put out in the next year or early next year. So for me, that's also quite interesting because for anybody who wants to get involved in studying microRNAs and reads a review paper and assumes that this is the dogma of how microRNAs are always generated, it's not really always the case. There's a lot of things that are still unknown about how this particular pathway actually works. And so we've also seen situations, and I think we have a few different cases of this now, where we see isoforms or variants of a canonical microRNA, and the abundance of that particular isoform appearing in isolates from glioblastoma specimens. So this is telling me that it's not just that a particular microRNA is being affected, that there are other components at the protein level that regulate microRNA activity uh, are sort of being regulated in a full-blown cancerous state. And so we want to clearly understand what are the initiating steps that happen in a almost like a precancerous lesion, if you want to compare it to a colon cancer system as an example, and really understand what are the early determinants that affect microRNA processing. So that's kind of our goal there. And what I would like to just say, though, back to so for us, just to sort of blindly go into this process, it seems kind of naive to do. So in circling back to the inflammatory state, what we already know, and this is from a review we had just published, and we have some of our own uh, data as well with some additional microRNAs that clearly seem to be differentially expressed, in some cases elevated, in some cases down regulated under states of inflammation. So whether you could take some certain types of cells and treat them with a bunch of cytokine, you can see changes in expression profile of microRNAs when you culture like astrocytes in a dish. Versus sometimes from other investigators, when they've done in vivo models, they can see sort of systemic wide changes in cytokine production when there's the sort of onset of a brain tumor xenograft in that mouse model system. And so this also is important to know because to mention because it's not just in the cancerous state that we find these microRNAs being differentially expressed. It is also resulting in sort of other neurologic behaviors or neurologic disorders that you can see. So something like I'll pick up miR-155 as an example, as a classic microRNA that seems to be predominant in macrophages, and it's not just linked with cancer or regulating cancer growth. It's actually really important in modulating or fine-tuning the neuroinflammatory state and sort of regulating whether or not an individual is going to be experiencing some phenotypes akin to neurodegeneration. So um, again, my philosophy still for my group and my lab is still at some point to even understand what's going on in a cancerous state, we sometimes have to step back and really understand what is the normal function of something like a microRNA that can have many, many gene targets. And as that sort of normal homeostatic state starts to be tipped to uh, a state of a precancerous lesion or precancerous state, what is going on with both the microRNA expression level and what is going on with the messenger RNA, but also now what is going on with the suite of other non-coding RNAs, including the circ RNAs and long non-coding RNAs. And what we're trying to do, I think, with some others as well, building an entire interactome where we can tease apart sort of these massive, uh, non-coding RNA networks and really start pinpointing what may be the hopefully an initiator event, something akin to like that we could develop into a therapy like a PDL1 therapy from the non-coding RNA side, but that we would know that it's going to be linked with controlling uh, a whole pleiotropic suite of sort of effects that are related to neuroinflammation, which will have multifaceted effects. And so it could be one of these, a few of these microRNAs could have very, very strong response when coupled with an additional therapy like another immune modulator, for instance. So we're trying to be a little bit smarter when we are developing our quote unquote microRNA drug targets, right? And so we're trying to let the neuroinflammation kind of field kind of guide us in that approach. And I thought I was kind of silly at first to take this approach, but I didn't have enough time to get all of my slide deck together. But we have started doing some array profiling, and we've actually started treating some astrocytes with different drugs. And I'm not going to have time to go into this right now, but we do actually see the left-hand side of this array map is cells that are kind of treated with certain conditions, but none of them containing any kind of cytokine. And the panel off on the red over here is actually data that is they're being treated with a whole uh, sort of not just one cytokine, but a few different ones. So we're kind of normalizing some data based on experimental conditions, but we can clearly see that we're getting a pretty robust response of microRNAs that will be elevated in astrocytes when you treat them under certain uh, neuroinflammatory stimulatory conditions. And we can actually alter at least the growth of those astrocytes, which is actually being shown on the right-hand panel, based on the presence of just the microRNA alone. So what we're trying to say is that sometimes the abundance of the microRNA can kind of recapitulate a neuroinflammatory state. Um, and so we like this model. We've actually gotten some really good uh, preliminary data from this. And I think the reason why I got invited to give this webinar and the reason why I accepted most likely, and I'm very humble for this, is to begin interacting with the members of this group. And much as our previous speaker had indicated, perhaps try to form some kind of research collaboration and test some of these moieties and try to get some grant funding for this work because the non-coding RNA field again started when I was probably in college, so an entire lifetime, but not a full lifetime yet. You've seen us understand how a non-coding RNA from the very most biochemical level just sort of interacts with another messenger RNA up to now where we clearly have a whole wide variety of uh, tools, the diagnostic capabilities to find these microRNAs, other non-coding RNAs, understand the regulatory potential of these entities, but also start developing smart therapeutics to introduce non-coding RNA into a mammalian system and try to get it targeted to a particular tissue. And I think this is going to be one of our challenges especially in the brain cancer space, trying to get big, bulky, charged RNA molecule across what's presumably thought as a protected organ and get a sufficient dose of or the payload basically of our non-coding RNA into those cell types. So this was just a slide just to kind of show the audience that, there's a whole field that's been dedicated to modifying RNA. This is really more of a chemistry-based approach, but please be careful if anybody does work in the non-coding RNA space. An integumentary is actually a very specific thing. It's a cholesterol molecule that's attached onto an anti-microRNA. Not all inhibitors of a microRNA are quote unquote integumentars. A lot of the newer methodologies, we 2-O-methylate them. We put 2-fluoro markers on them and things like this. We make sometimes LNA, DNA mixmers. So basically the chemistry seems kind of maybe from a biological standpoint, it seems kind of simple, but it's actually difficult to really bulk make some of these therapeutic entities. And then I think again, it's really to sort of echo some of the other points from the other speakers. I really do actually think it's a limitation of sometimes some funding because from our side, from a biochemical standpoint, um, we would love to test all these modifications and add on all of these sort of adapters and see how well they hone to different tissue sites. But there's just not enough funding to kind of test all different permutations. So I think this is still a little bit where a bottleneck may be in our field. And this is where we're still kind of seeking out uh, collaborations with other individuals or whoever wants to work on this kind of project to see if their kind of bioengineered entity can kind of deliver non-coding any kind of non-coding RNA to a target organ site. Um, I think that's where what our goal is. So I think I'm gonna, for the sake of time, I don't know, I think I'm right on time actually. I'm probably gonna end and just again thank everyone who has invited us into this fold. I think it's a great audience, and this is a great field to go into, and we're definitely looking for uh, collaborators from all different areas to kind of tackle this problem. Our goal is to really change this curve from getting a 50% mortality rate in over a few years to getting a bulk of these patients to be living beyond uh, the 10-year mark. So we've been at this for about 6 or 7 years now, and we've gotten somewhat far. So I think it's just a matter of really continued support from the academic community and uh, testing some of our candidate molecules. But yeah, no, thank you very much for the time, and I'm happy to answer any questions.